In this video, I'm going to talk about the absorption of iron. And so this occurs mostly in the small intestine and quite a bit of it in the duodenum at the brush border. And so I'll cover some of the mechanisms here for absorption and control of absorption because there are quite a few controls for absorption of iron since, we obviously need it. It's of the trace minerals, it's the one that we have the most of, but we don't want too much of it because it can become toxic. And there are ways of actually sort of sequestering it so that pathogens don't get it. And, I'm going to cover that in a future video about what's called nutritional immunity. And so that's the way that we sort of sequester different, mostly these electrolytes away from pathogens so that we can essentially try to starve the pathogens of things like iron. And so here is some of the major pathways for absorbing iron. And there's uh, essentially two. And there's the one for absorbing iron that's in heme groups, and then there is this one here down here, which is for absorbing labile iron, so iron that's not bound to another protein or large molecule or anything. And so we see that this one up here with the heme is taken into the cell from the intestinal lumen by this HCP1 here, which that is the heme carrier protein 1. And so then that kind of comes in here, and we see that this cytochrome P450 here, so this HO1 cytochrome P450, which is the cytosolic heme oxygenase 1 NADPH cytochrome P450 reductase, which is a bit of a mouthful, so that's why it's shortened to that. But that catabolizes the heme into biliverdin and carbon monoxide and releases the iron 2. And so that's the thing to notice down here is that it's the iron 2 plus that we actually absorb. So this iron 3 plus, this more oxidized state of it, is actually not very soluble. And so it has to be reduced by this duodenal cytochrome B right here. And so that is this one right here, the duodenal cytochrome B. So it goes from the Fe3 plus to the Fe2 plus, and then this DMT1, so that's the divalent metal transporter, brings that in. And then this figure here is showing this PCBP2, which is a chaperone protein, actually brings the Fe2 plus down here to this FPN1, where this FPN1 is what actually well, not sort of, it is the protein that actually puts the iron into the bloodstream here. Up here we have this happening with the FLVCR and the ABCG2 up here with the heme. Down here it primarily happens with this FPN1 right here. So then there is this RTF here, which that is the transferrin receptor. And so transferrin is a protein that's produced in the liver that regulates absorption of iron into the blood. And so there is this other pathway here, or this sort of regulation of iron going into the blood from these enterocytes here. And so, like I said, that is uh, iron is sort of highly controlled for its absorption here because we don't want too much or too little. The other thing to notice here, we have this ferritin right here, which is sort of a protein that stores iron, and we'll actually look at the structure of that here in a little bit. All right, so this is just kind of a more simplified version of the non-heme pathway here. So once again, we have the Fe3 plus going to the Fe2 plus with the duodenal cytochrome B here. Then the DMT1 here is actually absorbing it. Then this ferroportin here, which is that FPN1, is then sending it here into the bloodstream. And so this DCYTB, this duodenal cytochrome B, so as I said, that reduces it from the Fe3 plus to the Fe2 plus, and that actually does it with the help of this molecule here, which is vitamin C. And so vitamin C is actually needed for this reduction. And so we can actually look at the structure of this right here. So we see that it's this homodimer here. And if we sort of zoom in onto this side, we see that we have these hemes here, which are what will hold on to the iron when it's actually on these proteins here. And then up here in orange, I actually have this is the vitamin C molecule here in orange. We can see what I have here in pink is what's holding the vitamin C in there. Down here in yellow are the residues that are holding onto the heme there. And then we have these channel residues right here, this tyrosine and this phenylalanine here. Like I said, we can see that as a homodimer, so we still have these hemes here, and we have the vitamin C over on the other side as well. So we can see that that is the protein that is doing this this sorbate, which is vitamin C ferridoreductase, so changing the Fe3 plus to the Fe2 plus. All right, down here we can look at this in terms of iron deficiency, also in hypoxia, and iron loading, which is when we have a lot of iron going on. So in iron deficiency, we actually have these HIF2 alpha here, which are transcription factors, and so they actually bind to the DNA in the nucleus and actually increase well, it upregulates, it increases the expression of the DCYTB, the DMT1, and the FPN1. And so these HIF2 alphas are sort of sensing that we have an iron deficiency, and so it actually upregulates those in order to try and get more iron into the cell. And so then we have, I think it was this PhD, yeah, these PhD 1 through 3 here, and these actually sort of sense if we have too much iron, so if we have iron loading. And so that we see that that inhibits the HIF2 alpha here, which reduces the amount of the duodenal cytochrome B, the DMT1 and the FPN1 here. And so we'll actually look at this hepcidin here also in a little bit. Yeah, and so this is just the description of kind of what I was saying up there, where we have this HIF2 alpha is increasing the DCYTB, the DMT1, and the FPN1 here. And then we have this well, it's that this HIF2 alpha is actually being controlled at the post-translational level by binding to the 5' UTR there. So this is that DMT1, the divalent metal transporter 1, and this is the proposed mechanism from a paper. It proposes a few mechanisms, but in the end it sort of says that this is the model that kind of fits the data the best. And so we see we have these Fe2 plus outside in the in the intestinal luma there, and that binds onto this DMT1 here, which then undergoes a conformational change so that it can let the iron out right there, and so that lets it out inside there. And we have this going on here where we can cause this to actually pinch off and become a vesicle here. And so that can actually happen so that we can store iron in these vesicles here and then release and take those up inside the cytoplasm. And I put this here because like I said, this is called the divalent metal transporter, and we're talking about it here in terms of transporting iron, but it can also transport these divalent ions here, so manganese, cobalt, zinc, copper, and lead. So it can be used to transport a lot of those different things. And so this is just some kinetics work here, looking at the DMT1, and it's looking. This one was specifically looking at this histidine at the 272 position, and we see that if that is sort of mutated to this alanine, which is kind of a standard mutation when doing these to sort of shut off that specific residue, to sort of actually look at what that specific residue is up to, and we see that that reduces the kinetics here, that it reduces the turnover rate by by a pretty large factor here, and so that's showing that that is an important residue in here. But anyway, then I also have this over here, which I thought was interesting, especially right here, because it's just showing that this DMT1 works best at this relatively low pH, which when you think about it, makes kind of sense a little bit, because this is occurring in the duodenum, where you're getting that gastric juice emptied out of the stomach, and it is being neutralized a bit in the duodenum, but it's still going to be somewhat acidic, and so it makes sense that it would have the higher activity at this lower pH here. And so this is the description of it here. And once again, these figures are all things that I did not make. I took these from papers, and those will be linked to in the description down below if you wanted to look at these a little bit more in depth on your own. All right, then we can look at this ferroportin here, which is what transports the iron from in the enterocyte into the bloodstream. So this is saying that with low iron, we have this sort of activated so that it can actually transport iron, but at high iron, we have this hepcidin here, which actually binds to this transporter here and shuts it off. So over here we have the kinetics looking at the transport of iron and the transport of cobalt. We see here, well, these are the actual time series here for those kinetics right there. But then looking at the different concentrations, we can look at that right here, and we see that this has the turnover rate going up here, and we can see that it's fairly tight binding. So a lot of these transporters here appear to be fairly slow moving, but they are very tight binding, and so that kind of makes sense because again, we want that tight control of the iron absorption, because we don't want getting too much of it into our bloodstream, because that can become toxic. So I actually have the ferroportin structure. We can look at that right here. So green is the ferroportin protein. This thing in yellow here is that hepcidin right there. So this little thing right here in yellow is the actual hepcidin. And so in this one, these sort of salmon colored spheres here are actually cobalt atoms, but those would also be where the iron atoms would bind to it. And I have kind of in this purplish color the residues that are binding to those metal ions. And so yeah, that is where those bind to. And one other thing I forgot to mention is actually that the DMT1, like I said, it happens in this lower pH. And so one of the things that actually determines that I forgot to mention is that it is a iron and H plus co-transporter. And so it requires an influx of, well, it brings iron in, but it does that by bringing H plus, so protons, acid essentially, down the gradient. So there's more acid in the intestinal lumen than there is inside the cell, and so it kind of uses the energy of the H pluses going down that potential into the cells to actually power the movement of iron into the cell. But anyway, I just wanted to make sure I mentioned that because that's kind of an important way of how the DMT1 works. But yeah, anyway, the ferroportin is sort of regulated by this hepcidin, but also, as we saw above, by that HIF2 alpha, the HIF2 alpha, which sort of is a transcription factor that controls the expression of the ferroportin. So the other thing I wanted to show, so remember up here I talked about this ferritin, and so I have the structure for that here. So we can see this is kind of a large sort of spherical protein here made out of multiple subunits. So I have here in green just one of the subunits for that. And all these orange spheres here are iron. So this was taken, I believe, when it was soaked in iron for 15 minutes. And so we see that this has taken up a lot of iron, so it's storing all that iron in there. These salmon colored ones, if I remember correctly, yeah, those are cadmium. So these ones up here, these CD, those are cadmium. And so I have on here on this one subunit, so we can see these irons here, and we see that they are coordinated to these residues that I have here in magenta. And so yeah, this, like I said, is the ferritin, and it is storing the iron. And just like with the papers that I take figures from, I also leave in the description down below the PDB numbers or PDB codes for all of the structures I use in case you wanted to look at those a little bit more in detail on your own as well. But yeah, that is the ferritin there, which is what's shown here with all these irons binding to it. Yeah, so anyway, the sort of take-home message here, we have this heme pathway, so this HCP1 takes that in. It can either get broken down by this cytochrome P450 into this labile iron here, which can then sort of bind to the ferritin there, or it can bind to this PCBP2 chaperone protein and then get carried into the blood by the FPN1, or it can continue on into the blood as heme and get transported into the blood by this FLVCR or this ABCG2. Or if we have the iron here that's not bound to heme in the blood, it has to be reduced by this DCYTB into Fe2+, and then it's taken in by the DMT1, where then this PCBP2 chaperones it over to the FPN1, and so then that transports it into the blood. And I'm going to go through like a lot of different stuff and the absorption for a lot of different stuff, but after I finish that, that's when I'll actually get to sort of the transport of all these ions and all the vitamins and macromolecules and things like that in the blood. So that will be kind of the next section after I get through all of these absorption videos. But anyway, I hope you found this video helpful, and I will see you in the next one.